trees take several years for the initiation of flowering, whereas an annual herb flowers within few months, which means each plant requires a specific time period to complete their vegetative phase, which is followed by the reproductive phase. So, the flowering are controlled by light period called photoperiodism. Flowering is controlled by light period called photoperiodism, and also it is controlled by temperature called vernalization. First, we are discussing the flowering controlled by a light period called photoperiodism. Photoperiodism is a physiological change on flowering due to the relative length of light and darkness. So, the flowering is due to the relative length of light and darkness. So, the flowering is depend upon the relative length of light and darkness. It's called photoperiodism. The photoperiod required to induce flowering is called critical day length. The photoperiod required to induce flowering is called critical day length. Maryland mammoth, it is a tobacco variety. It requires 12 hours of light for flowering, whereas cocklebear, it's Sandium pennsylvanium, it requires 15.05 hours of light for flowering. That is, each plant it has a variable in their own way for the flowering. It takes difference in the critical day length for flowering. Based on the photoperiodism, plants can be classified as 3 types. Long day plants, short day plants, and 3 day neutral plants. Long day plants, short day plants, and day neutral plants. Long day plants are simply the plants that require long critical day length for flowering. The plants that require long critical day length for flowering. That is, it requires large amount of light. Then, short day plants are the plants that require short critical day length for flowering. That is, it requires less amount of light for flowering. So, last one is day neutral plants. It is the word itself, it is clear that the plants can flower in all possible periods, photoperiods. Example for long day plants are barley, pea, oats, whereas short day plants are tobacco, cocklebear, rice, chrysanthemum, soybean. Day neutral plants are potato, rhododendron, tomato, and cotton. So these are based on the photoperiodism. Plants are classified as 3 types. Then we are going to photoperiodic induction. What is photoperiodic induction? So, an appropriate photoperiod in 24-hour cycle constitutes one inductive cycle. So one inductive cycle, which means 24-hour cycle, constitutes one inductive cycle. Plants may require one or more inductive cycles for flowering. The phenomena of conversion of leaf primordia to flower primordia under the influence of suitable inductive cycle is called photoperiodic induction. Photoperiodic induction. of leaf primordia into flower primordia under the influence of inductive cycle. This is called photoperiodic induction. Different plants may have different photoperiodic induction. Xanthemum, it is a short day plant. It requires one inductive cycle for flowering, whereas Plantago, it is a long day plant. It required 25 inductive cycles for flowering. These are all about photoperiodic induction. Then the site of photoinductive perception. Site of photoinductive perception. Site for photoinductive perception. The photoperiodic stimulus. The photoperiodic stimulus is received by leaves. The photoperiodic stimulus is received by leaves. The floral hormone is synthesized in leaves and is translocated to the apical tip. So, leaves also play a significant role in flowering. So, the floral hormone, the flowering hormone called floragen. It's a flowering hormone. It is synthesized in the leaves. The floral hormone is synthesized in the leaves and translocated to the apical tip to promote flowering. This is called the photoinductive perceptions. It can be well explained by using an experiment. The experiment is done using cockleber, that is Xanthium pennsylvania. What they done is, we know that this is a short day plant. It's a short day plant. What they actually done using this experiment is, they defoliate the plant, that is no leaves. They defoliate the plants and they place the plant in short day condition. Short day condition. The result is no flower. The result is no flower. So, they defoliate the xanthium and they place the defoliated plant in short day condition. The result is no flowering. Then they done same. No leaves, that is defoliated plant kept in long day condition. You must aware that it is a short day plant, but they kept the defoliated plant in long day condition. The result is also no flower. Then what they done is, they kept only one leaf. They kept one leaf and they kept one leaf in the plants and give the short day condition. They kept the short day condition. Flowering occurs. Flowering occurs. Again, they what done is, they kept one flower. Sorry, they kept one leaf and they give the short day condition only for one leaf, and rest of the leaves of the plant gives long day condition. Again, the flowering occurs. So, from this, what it means that the photoperiodic stimulus is perceived by the leaves and they are responsible for the transport of the floral hormones to the apical tip, which promote flowering. So, you can go through the experiment. It is in your textbook. The importance of photoperiodism is, it helps in hybridization experiment. photoperiodism of a particular plant, it helps in the hybridization experiment of that particular plant. It helps in the hybridization of that particular plant. And also it is an excellent example that the external factor, using the external factors, the physiological changes may happen or happens in the plants. That is, external factor induces the physiological changes in the plant. Now we can discuss what is phytohormone. Phytohormone is simply bluish bubbly protein. Phytochrome. Phytochrome is a bluish billy protein pigment. Bluish billy protein pigment, and it is responsible for the perception of light in photophysiological process. So the phytochrome exists in two interconvertible forms, that is PR and PFR. The PR form is the pigment that absorbs red light, whereas PFR form is the pigment that absorbs far red light. The PR, it converted to PFR by absorbing red light of 660 nanometer. The PR absorbs red light of wavelength 660 nanometer and become PFR. And the PFR is interconvertible to PR by absorbing far red light of 730 nanometer and become PR. So here the PR is biologically inactive, but it is the stable form, whereas the PFR is biologically active but highly unstable form. So these are the two interconvertible phytochrome, PR and PFR. The PR is help in short day plants. What they are doing is the PR promote flowering. In short day plants, the PR promote flowering, whereas PFR inhibit flowering. In long day plants, the PFR helps in flowering, that is promote flowering, whereas the PR form inhibit flowering. So these are about the photoperiodism. Okay, then vernalization. Besides photoperiod, certain plants requires a low temperature for flowering. So flowering requires temperature. Certain temperature is required for the induction of flowering, is called vernalization. Many species of biennials and perennials are induced to flowering by low temperature exposure, that is 0 degrees Celsius to 5 degrees Celsius, that is low temperature. Low temperature which induces flowering. This process is called vernalization. The mechanism of vernalization can be explained using two theories: hypothesis of phasic development and hypothesis of hormonal involvement. So the mechanism of vernalization can be explained using two theories. First one is hypothesis of phase development and second one is hypothesis of hormonal involvement. Hypothesis of phase development and hypothesis of hormonal involvement. hypothesis of phase development. For an annual seed plant, there is two stages. First one is thermostage and second one is photostage. In the annual seed plant has two stages in their growth. First one is thermostage and second one is photostage. In thermostage, the vegetative phase requires low temperature and moisture. Low temperature and moist. So the vegetative phase of the annual seed plant requires low temperature and moist. It's called the thermostage, and in photostage it requires high temperature. The plant requires high temperature for the production of the floral hormone called phloriston. So these are the hypothesis of phasic development, whereas the hypothesis of hormonal development or hormonal involvement involvement. What it says that according to Purvis, scientist Purvis, what he notified that now, A is a substance which is formed from a precursor molecule. A is a substance which is formed from a precursor molecule, and this A is converted to B. This A is converted to B after chilling. And this B is highly unstable. So, at a suitable temperature, the B is converted to a stable compound called D. This is unstable. At suitable temperature, it may convert it to a stable compound D. It is called Vernalization. Stable compound B called Vernalization. And this Vernalization is converted to F called phloriston. Phloriston is the hormone which is responsible for flowering, and this phloriston is transported to tip of the plant, that is shoot tip of the plant. So from precursor molecule, a substance A is formed, and under chilling, the A converted to an unstable compound called B. In suitable temperature, the B is converted to a stable compound called D, also called as Vernalization, and this Vernalization converted to phloriston, and phloriston is transported to the plant tip for flowering. And this D under high temperature, may convert it to C, and this C undergo devernalization. So this is about hormonal involvement hypothesis. Devernalization is nothing but it's simply the reversal of vernalization. Now, come to the practical application of vernalization. Vernalization shortens the vegetative periods and induces the plant to flower. Vernalization induce flowering. Vernalization may shortens or they ceases the vegetative growth, and it induces flowering in plants and it increases the cold resistance of plant. It increases cold resistance in plants. It increases the cold resistance in plants and also it increases the resistance of plants to fungal disease. Also it increases the resistance to fungal disease. Resistance to fungal disease. Also, plant breeding can be accelerated using vernalization techniques. So these are all about vernalization. Next topic is seed germination. Seed germination is nothing but the activation and growth of embryo from seed into seedlings during favorable conditions. So the growth of the activation and growth of embryo from seed to seedling by the growth and activation of embryo from seed to seedling under favorable condition is called seed germination. The seed germination are of two methods. I think you all are familiar with these methods. First one is apigeal germination. First one is apigeal germination and the second one is hypogeal germination. Apigeal germination and hypogeal germination. In apigeal germination, the cotyledons are pushed out of the soil, whereas in hypogeal germination, the cotyledons are buried inside the soil. Here, cotyledons push out of soil, whereas in hypogeal germination, the cotyledons remains buried in the soil. So apigeal germination means the cotyledon pushes out of the soil, whereas hypogeal germination means cotyledon remains buried in the soil. In apigeal germination, this happens due to the elongation of hypocotyl, whereas in hypogeal germination, this happens due to the elongation of epicotyls. So these are about the two different types of germination and the examples are given in your textbook. Castor and bean are example of apigeal germination, whereas maize, peas, etc. are hypogeal germination. Then factors affecting germination. What are the factors affecting germination? There are internal factors as well as external factors that affect germination. First we can look the external factors that affect the germination. External factors that affect the germination. What are the external factors? The first main factor is water. Water is the first external factors. What it has done in seed germination is, when the seed gets sufficient amount of water, it activates the enzymes inside the seeds which may digest the reserve food materials, which leads to the germination of seeds. So when seed soak or seed imbibes water, the enzymes activate and these enzymes may digest the reserve food materials inside the seeds, leads to the seed germination. The second external factor is temperature. The second external factor is temperature. So the seeds fail to germinate at very low temperature and at very high temperature. So an optimum temperature is required for the germination of seeds. So optimum temperature in the sense 25 degrees Celsius to 35 degrees Celsius is 25 degrees Celsius to 35 degrees Celsius is an optimum temperature which is required for the germination of seeds. And the external factors that affect the germination is oxygen. For seeds, aerobic respiration is required for the germination. So, oxygen is necessary for the germination of seeds. Then, light. There are many photoblastic seeds, that is photosensitive seeds. It requires light for the germination. It requires light for the germination. It requires a particular amount of light for the germination. Those seeds are called photoblastic seeds. Then last one is soil conditions. Germination of seed in its natural habit is influenced by soil condition. The rest of them can be influenced by while we are performing some lab experiments. But the soil condition is, if a seed is germinating in its natural conditions, so soil conditions may influence the germination of those seeds, like water holding capacity, mineral composition and aeration of the soil. So these are the external factors that affect the germination of seeds. Now going to the internal factors. Going to the internal factors. First one is maturity of embryo. Maturity of embryo. Some plants may contain immature embryos. Some plant seeds may contain immature embryos which may affect the germination of seeds. So such seeds may germinate only after the embryo become mature. So only the germination takes place when the embryo become mature stage. Then viability. Second internal factor is viability. So usually seeds remain viable for a particular period. The viability of seeds may differ for different plant groups. The seed can be viable from few days to more than 100 years. So the viability plays an important role in seed germination. So seed germinate only within that viability period. The third one is dormancy. So the third one is dormancy. Seeds of many plants are dormant at the time of shedding. So dormancy, about dormancy we are going to study in detail. So these are the internal factors that affect seed germination. So these are about seed germination. Now we are going to take seed dormancy. Seed dormancy is nothing but it is a condition of seed when it fails to germinate even in suitable condition. That is, a seed have it's all suitable, favorable condition for germination. Even though there is favorable condition, some seeds are failed to germinate. This condition is called seed dormancy. Seed dormancy. There are two main reasons for the development of dormancy. First one is imposed dormancy. Second one is innate dormancy. So, the reason for dormancy can be imposed dormancy or innate dormancy. Imposed dormancy is due to low moisture and low temperature. It is due to low temperature and moisture. So, the imposed dormancy is due to low temperature and moisture, whereas innate dormancy is related to the properties of seed itself. So, if there is any problems in the seed itself which leads to dormancy, it is called innate dormancy, whereas the external factors like low temperature and moisture is reason for the dormancy, it is called imposed dormancy. Then what are the factors causing dormancy of seed? Factors causing dormancy. The first one may be hard, tough seed coat. Dormancy can be due to the seed coat may be hard or tough. Hard, tough seed coat. It is a reason, one of the reason for seed dormancy. The second reason is imperfectly developed embryos. The imperfectly developed embryos are called rudimentary embryos, which promotes dormancy. The imperfectly developed embryos are called rudimentary embryo. So it is also a reason for seed dormancy. Then lack of light. Lack of light and a lack of temperature. So lack of light, temperature, moisture, etc. Some external factors also affect the seed to germinate. That is, it's the reason for seed dormancy. Then other factor is the presence of inhibitors like phenolic compounds. Some inhibitors like phenolic compounds may cause seed dormancy. So these are the factors that affect seed dormancy. Now the method of breaking dormancy. How can we break dormancy? What are the methods of breaking dormancy? Methods of breaking dormancy. What are the methods of breaking dormancy? First one is scarification. Scarification. It is the mechanical and chemical treatments like cutting or chipping of the hard tough seed coat and uses of organic solvent to remove waxy or fatty compounds are called scarification. That is, the hard seed coat is removed mechanically using cutting or chopping, or some organic solvents to remove the waxy and fatty compounds on the seed coat. This method is called scarification. The second method is called impaction. The second method is called impaction. In some seeds, water and oxygen are unable to penetrate to the microbial due to the blockages by coxel. So, we know that the water and oxygen should be reached inside the embryo for the development of embryo and seed germination. So, due to the blockage in microbial, the oxygen and sufficient amount of water cannot be reached there. This problem can be caused by the seeds are shaken vigorously to remove the block. Impaction means shaking. So, seeds can be shaken vigorously to remove this block. The third one is stratification. The third one is stratification. That is, seeds of Rosaceae family plants will not germinate until they have been exposed to well-aerated moist condition under low temperature for weeks to months. That is, the plants under Rosaceae family, they will not germinate until they require a well-aerated moist condition under low temperature 0 to 10 degrees Celsius under low temperature for many weeks to months. Many weeks to months. This is called stratification. And the fourth one is alternating temperature. Alternating temperature. Germination of some seeds is strongly promoted by alternating daily temperatures. So, an alternation of low and high temperature improves the germination of seeds. So, in alternating some plant seeds requires alternating temperature. So, alternating low and high temperature is required for the germination of seed. Then, last one is light. Light. The dormancy of photoplastic seeds can be broken by exposing them to red light. We already discussed about the photoplastic seeds. That is, photoplastic seeds are seeds that requires a sufficient amount of light for germination. They are called photoplastic seeds. So, the dormancy of photoplastic seeds can be recovered by exposing them to red light. So, which breaks the dormancy and induces seed germination. So, these are methods of breaking seed dormancy: scarification, impaction, stratification, alternating temperature and light. So, next topic is senescence. We know that plant life comprises some sequential events like germination, juvenile stage, maturation, old age and death. Senescence is simply old age of plants. Old age plants are called senescent plants. The senescence refers to collective, progressive and deteriorating process which ultimately lead to complete loss of organization and function. At the senescent stage, the cell loses complete loss of organization and function. So, there is no function or organization may carried out in the senescent cells. The branch of botany which deals with aging, accession and senescence is called phytogermatology. Phytogermatology. So, the old age of plants are called senescence. Different types of senescence are types of senescence. First one, overall senescence. Overall senescence. Overall senescence, which means entire plant get affected and dies. The entire plant get affected and the plant get dies. Example, wheat, soybean, agave and bamboo. So these overall senescence may happen in both annual plants as well as perennial plants. So overall senescence means entire plant get affected and dies. The second one is top senescence. Second one is top senescence. Top senescence means the aerial part of the plants affected. The aerial part of the plant affected and senescent. It is top senescence. Example, banana, gladiolus, etc. The third one is deciduous senescence. Third one is deciduous senescence. It is common in deciduous plants and it occurs only in leaves of plants, bulk of stem and the root system remains alien. So, in deciduous plants, it is common in deciduous plants. Deciduous senescence means the leaves and bulk of the stem may affect, but the leaves of the plant may affect senescence, but the bulk and root system may remain alien. Then the last one is progressive senescence. Progressive senescence means it is a gradual senescence in which first it occurs in old leaves. The senescence occurs in old leaves followed by new leaves, then stem and finally root system. So it is common in annuals. The progressive senescence means senescence occur progressively. So these are the different types of senescence. Now going through the physiology of senescence. Going through the physiology of senescence, it is nothing but the cell undergo changes in structure. The structure of the cell undergo changes. Cell structure changes. Cell structure changes. Then vacuole of the cell act as lysosome and secrete hydrolytic enzymes. Several hydrolytic enzymes are produced. The starch content is decreased in the cell. Decreased starch content. Hydrolytic enzymes produce decreased starch content in the cell. Decreased starch content in the cell. Then the photosynthesis is reduced due to the accumulation of anthocyanin pigments. So the rate of photosynthesis decreases. Rate of photosynthesis decreases due to the accumulation of anthocyanin pigments, which results in the color changing of the leaf. Then there is a marked decrease in protein content in senescent organ. Decrease in protein content. Decrease in protein content. And also due to the activity of RNase, the rRNA level is decreased in cell. So due to the activity of RNase, the ribosomal RNA level decreases. RNA level decreases in ribosomal RNA level decreases in cell due to the activity of RNase, just like the activity of DNA is the DNA level also decreases due to the activity of DNAs. So this enzyme activity results in the decreased level of RNA as well as DNA in the senescent cell. So these are about the physiology of senescence. Now, going through the factors affecting senescence. What are the factors that affecting senescence? The phytohormones like ABA and ethylene accelerate senescence while auxin and cytokinin retard senescence. ABA and ethylene increases senescence, whereas auxin and cytokinin decrease or retard senescence. Then nitrogen deficiency increases senescence, whereas nitrogen supply retards senescence. Nitrogen deficiency increase senescence. Nitrogen deficiency increase senescence, whereas nitrogen supply may retard senescence. Then high temperature may accelerate senescence. High temperature may increase senescence, whereas low temperature may retard senescence. Senescence is also rapid in dark. In dark condition, senescence is high. Whereas in light condition, senescence is less. Then water stress leads to the accumulation of ABA, results in senescence. We already discussed that ABA results in increased senescence. So water stress leads to the accumulation of the stress hormone ABA, which results in increasing senescence. So these are about the factors of senescence. Now, moving to programmed cell death. PCD, programmed cell death. Programmed cell death is simply the senescence of an individual cell. The senescence of an individual cell is programmed cell death. The proteolytic enzymes involving PCD is phytapsase in plants and campsase in animals. Phytapsase in plants and caspase in animals. So these enzymes are responsible for the programmed cell death in animals as well as in plants. The nutrients and other substrate from senescent cells are remobilized or relocated to other part of the plants. That is, all the nutrients and other substrates from the senescent cell is transported or remobilized or relocated to other tissues for their survival. And the protoplasm of developing xylem and tracheids die and disappear at the maturity to make them functionally efficient to conduct water for transport. So the programmed cell death of protoplasm of xylem vessels and tracheids. The programmed cell death of protoplasm of xylem vessels and tracheids helps in the water conduction at its maturity stage. And also in some aquatic plants, the aerochema is normally formed in different parts of the plant, such as roots and stem, which encloses large airspace that are created through programmed cell death. So the aerochema are situated in the intercellular spaces that is formed due to the programmed cell death. And in the development of unisexual flowers, actually we know that what are unisexual flowers? Are they male or female flowers? So in unisexual flower production, initially both male and female flowers are produced initially, but due to the programmed cell death process, one of the flowers may die. Either male or female flowers may occur. So the presence of unisexual flowers in the plants is due to the programmed cell death. Initially both male and female flowers are present at early stages, but only one of these two completes its development and the other will abort due to programmed cell death. So these are about programmed cell death. Okay students, we are moving to the final topic of this chapter, abscission. Abscission is a physiological process of shedding organs like leaves, flowers, fruits and seeds from the parent plant body. When these parts are removed, the plant seal off its vascular system to prevent the loss of water and nutrients. So simply, abscission is the final stage of senescence. Abscission is the final stage of senescence. So after senescence, the senescent part will be shed off from the parent body. We know that in temperate regions, all the leaves of deciduous plants fall in autumn and give rise to a naked appearance. Then the new leaves are developed in the subsequent spring seasons. This is due to the process abscission. But in evergreen plants, this is quite different. In evergreen plants, the abscission is not uniform. The abscission is gradual. So, in evergreen plants, the older leaves fall off while new leaves are developed continuously throughout the year. That's why always the evergreen plants looks like leafy plants, whereas the deciduous plants in the autumn season, all the leaves are shed off, and again when the spring season comes, all the new leaves are came. This is due to abscission. Now we can look to the morphological and anatomical changes during abscission. What are the morphological and anatomical changes occurs during abscission? Leaf abscission takes place at the base of petiole. The leaf abscission may take place at the base of the petiole. And it produces a layer called abscission layer. Abscission layer. Absorption layer is greenish gray in color, and it is formed of a row of cells which is arranged transversely. They are thin walled cells arranged transversely by a row of 2 to 15 cells thick. So this is the absorption layer. Here what the absorption going to take place. The cells of the absorption layer. Here the conductive systems are going. The cells in the absorption layer what they are doing is the cells of the absorption layer separates. The cells may separate due to the dissolution of middle lamella and primary walls by the activity of two enzymes called pectinase and cellulase. So, due to the activity of pectinase and cellulase, the cell in the absorption layer may separate. The cell in the absorption layer may separate due to the dissolution of middle lamella of the cells. This is due to the activity of two enzymes, pectinase and cellulase. So this results in the loosening of cells in the absorption layer. Along with that, tyloses are also formed blocking the conducting system. If you consider this is the conducting system, tylose are some bellow-like structures which is formed in the conducting system, which may result in the blockage of the conducting system. So, furthermore, transport of water, minerals or anything to that same part. These balloon-like structures are called tyloses. Degradation of chlorophyll occurred, leading to the change in color of leaves. So, the senescent part here, the chlorophyll molecules undergo degradation, which leads to change in the color of the part. So, the leaf detachment from the plant and the leaf falls. So, due to the degradation of chlorophylls in the leaf leads to change in the color of leaves, as well as the leaves detach and the leaves fall off. After absorption, outer layer of cell becomes suberized by the development of periodum. So after absorption, the outer layer of the cells becomes suberized by the activity or by the development of periodum. So this is the morphological and anatomical changes during absorption. Now we are looking the hormones influencing absorption. What are the hormones that influence absorption? The hormones influence absorption. Abscisic acid and ethylene. Abscisic acid and ethylene are the two hormones that induces absorption. Abscisic acid and ethylene are the two hormones that induces, which means it promotes absorption. Now, looking to the significance of absorption. What are the significance of absorption means? Significance of absorption. First one, the absorption separate dead parts of the plant like old leaves and dry roots. So it separates dead parts from the parent bovine. And the second one is it helps in the dispersal of fruits and continuing the life cycle of plants. Helps in dispersal of fruit. It helps in dispersal of fruit. Absorption of leaves in deciduous plants helps in water conservation during summer. So, the leaf absorption in that deciduous plants which helps in the water conservation during summer. So, it helps in water conservation. And the last significance is in lower plants, the shedding of vegetative parts like germe or plantlets help in vegetative propagation. We know that in lower plants the reproduction takes place through vegetative propagation. So the shaping of the vegetative parts like germe or plantlets helps in the vegetative reproduction. So these are about the significance of absorption. So these are about the chapter plant growth, plant development and growth. Okay, if you have any doubt, you can feel free to ask. Thank you.